Hello and welcome to week 2. Week 2 will be covering chapters 4 and 5. Chapter 4 is about the states of consciousness, and chapter 5 is about sensation and perception. First, we're going to be going through the 2 PowerPoints. During the 2 PowerPoints, I'll also be providing insight into your chapter questions for this week on the 2 chapters. So we're going to click on this link here in order to download chapter 4 PowerPoint, like so. So, chapter 4 is all about the states of consciousness. We want to understand what consciousness is, what it means to be conscious. We're going to learn about the altered states of consciousness during this chapter. Before we get started, I want you to think about and write down a few things. First, I want you to examine your state of awareness. Are you drowsy, alert? Are you intensely focused, or maybe daydreaming? Are you interested and absorbed in the context of what you're hearing? Or are you a little bit distracted? What do you think is influencing this feeling? Consider what you're aware of. Have you always been aware of what you're aware of? Or are you only now thinking about it because I'm asking? Now shift to your self-awareness. What's happening inside of you? How do you think you appear to others? What kind of mood are you in? These 3 questions and those things that I asked you to consider are things that we're going to look at as we study what consciousness is. Consciousness has been a favorite topic among psychologists for many years. William James, as I introduced in the first chapter, coined the term stream of consciousness. He did this to capture the moving, seemingly unbroken flow of conscious awareness. Freud then used the term consciousness to refer to aspects of the mind that could be retrieved voluntarily, in contrast to the unconscious part of the mind that remained hidden to voluntary searches for information. Contemporary cognitive psychologists debate the reasons why some information becomes conscious while other information does not. They ask big questions like, what does it mean to be conscious? Is consciousness uniquely human? What is the relationship between brain function and consciousness? Most of us would agree that bacteria do not process consciousness, but that people do. This implies that consciousness emerged at some point in the course of evolution. The exact location of that point remains highly debatable. The questions of the emergence of consciousness in animals is complicated by the multiple meanings of consciousness. Most, if not all, animals show variations in state of awareness. For example, with ducks, while they sleep, one hemisphere of their brain can process visual information from an eye that remains open. Birds positioned where they are most vulnerable to predators, such as at the end of a row, may spend twice as much time in this alert sleep than do birds in more protected positions. Now, in order to truly recognize if animals have consciousness is to look at the multiple meanings of consciousness. Question 1 for your chapter 4 questions is going to ask you to define what consciousness means to you. What does consciousness mean to you? And then to compare your definition to the standard definition you see here. Consciousness is an awareness of internal and external stimuli, such as feelings of hunger and pain or detection of light. Now, if that's what we're thinking about as the common definition, this is probably less common in the animal kingdom. Many animals would survive quite well by simply responding automatically to the world around them, without necessarily thinking, I smell a predator, or I smell food. The self-awareness aspect of consciousness, as exemplified in I statements, might be the rarest of all among living things. Now, sleep is often studied as a part of consciousness. Sleep is something that we all experience, is a quiet and mysterious pause in all of our daily lives. But it's helpful to study sleep when we are studying consciousness, because wakefulness is high levels of sensory awareness, thought, and behavior. In order to be conscious, we must also be experiencing wakefulness. Psychologists began studying biological rhythms, also known as circadian rhythms. A biological rhythm is an internal cycle of biological activity, which includes the fluctuation of body temperature, an individual's menstrual cycle, and your level of alertness. Circadian rhythm is the biological rhythm that occurs over approximately a 24-hour period of time. This is generated by the suprachiasmatic nucleus, or SCN. The sleep-wake cycle, one of our main circadian rhythms, is linked to our environment's natural light-dark cycle. The circadian rhythm is a built-in cycle of physical process in living beings. In humans, circadian rhythm can include your core body temperature, your alertness, your hormone productions, and other biological processes. And our sleep cycle is also greatly influenced by our biology. For example, if you were put in a cave and had no cues as to time, no watches, light, or clocks, your body would exhibit a natural rhythm of sleeping and waking that closely resembles the 24 to 25-hour cycle. You would instantly fall asleep at a natural time when sleep is typical. Now, the hypothalamus, which we learned about in chapter 3, controls our biological rhythm. It's responsible for maintaining that homeostasis. This is the tendency to maintain balance or optimal level within a biological system. This chart shows you circadian rhythms and the change in body temperature over 28 hours in a group of 8 young men. So body temperature rises throughout the waking day, peaking in the afternoon, and falls during sleep, with the lowest point occurring during the very early morning hours. Think about even in the hottest of hot days, you still tend to get a little chilly right before you wake up, the few hours before you wake up. And if we look deeper at the SCN, the suprachiasmatic nucleus, the sleep circadian rhythm is programmed by this group of brain cells in the hypothalamus. So the SCN is a group of brain cells within the hypothalamus, and it works very much like an internal clock, signaling our brain areas when to be aroused or awake to start the day and when to shut down or sleep for the day. The SCN is very responsive to light changes and takes its cues from your eyes. When your eyes transmit light information to the SCN within the hypothalamus, they are in essence telling it whether it's light or dark outside. The light information helps the SCN direct the release of melatonin, the hormone that facilitates sleep. Hence, light information and melatonin regulate your sleep circadian rhythm and help you get to sleep or awaken. When circadian rhythms are out of sync, the effect can be quite disruptive. Flying across time zones can leave you groggy and fatigued, also known as jet lag. It may take a day or more for your body's clock to align with this new time zone. People who work a rotating or unpredictable shifts tend to have lower productivity and more accidents than workers who have a consistent schedule. If we look deeper into melatonin and the sleep cycle, it's helpful for us to understand exactly what's happening. Our sleep-wake cycle is regulated by this particular hormone known as melatonin. Melatonin release is stimulated by darkness, making us sleepy, and inhibited by daylight. And this is released by the pineal gland. Sleep regulation is the brain's control of switching between sleep and wakefulness, as well as coordinating this cycle with the outside world. Each person has an individual circadian pattern of activity, known as a person's chronotype. So you might have a different circadian rhythm than that of a sibling or even a parent. Sleep regulation is your brain's control of switching between sleep and wakefulness. Now, disruptions of normal sleep. We discussed jet lag, symptoms resulting from the mismatch between our internal circadian cycles and our environment. We also talked about rotating shift work, a work schedule that changes from early to late on a daily, weekly basis. Devices like the light seen in this photo are designed to provide exposure to bright light to help people maintain a regular circadian rhythm. They can be helpful for people working night shifts or for people affected by seasonal variations in light. We'll learn more about that much later on in the term when we talk about seasonal affective disorder, where people can be very much so affected and their mental health can be affected strictly by the seasonal changes in light, when it gets darker earlier during the day. Now, sleep deprivation is another disruption to normal sleep. When someone is in sleep debt, this is a result of insufficient sleep on a chronic basis over long periods of time. A sleep rebound, however, is a sleep-deprived individual will tend to take a shorter time to fall asleep during subsequent opportunities for sleep. What you see in this figure illustrates some of the negative consequences of sleep deprivation. While cognitive deficits may be the most obvious, right? It takes you a little bit longer to retrieve things from your memory. You might have impaired judgment, loss of attention. Many body systems are also negatively impacted by lack of sleep. Many people didn't know that your immune system becomes impaired. So as you start to lose sleep, you also become less immune and more apt to get sick. I'm sure many of you would agree that you might get a cold during times when you're also sleep deprived. Your reaction time is less when you're sleep deprived. Your body doesn't have the ability to respond as quickly as it would like to. So if you're an athlete and play a sport and you're sleep deprived, you might not be able to run as fast as you'd like to or react as quickly as you'd like to during a play. Now, sleep needs oftentimes is based upon your age. You can see from this chart here that sleep needs at different ages does change. 0 to 3 months, 14 to 17 hours a day. So small infant children, they're merely sleeping and eating, and that's about it. Whereas if you get up to 18 to 25 years, you're looking at needing only 7 to 9 hours. 14 to 17 years, 8 to 10 hours. So think about this and maybe start to see how many hours of sleep you're getting yourself and where you fall in this chart. Now, What exactly is sleep? It's so important to us, obviously, but what is it? Sleep is a state marked by relatively low physical activity and a reduced sense of awareness. Sleep-wake cycles seem to be controlled by multiple brain areas, including the thalamus and the hypothalamus, and the pons. Sleep is associated with the secretion and regulation of many hormones, including melatonin, follicle-stimulating hormone, luteinizing hormone, and growth hormone. When your parents tried to convince you that it was time for bed because if you didn't go to bed, you wouldn't grow, they were not kidding. When you're asleep, many hormones are activated in your body that are actually stimulating health and wellness and development so that you can grow. Now, if we look deeper at the brain areas involved in sleep, the pituitary glands and the pineal glands, you see here, secrete a number of hormones during sleep. So this is where you see the growth hormone, the melatonin, and the other hormones that are secreted during sleep. Now, why are we sleeping? What's the purpose of sleep? First, it was discovered that it was an adaptive function. Okay? Sleep is essential to restore resources that are expended during the day. You need to rest. It's an evolutionary process. In order for us to survive and keep going, we need moments of rest and restoration. Sleep is an adaptive response to predatory risk, which increases in darkness. There is little evidence to support these explanations, though. We can't prove that we sleep in order to survive. Now, the cognitive function. So cognitive psychologists focused on sleep's importance for cognitive function and memory function. Research shows that sleep deprivation results in disruptions in cognition and memory deficits. These impairments become more severe as the amount of sleep deprivation increases. And they found that slow wave sleep appears to be essential for effective memory function. So it was the cognitive psychologists that said, yeah, we can think that this is we sleep for evolutionary purposes in order to survive. But really, we're doing it because in order for us to manage the memory and thoughts and knowledge we store in our brains, we need to rest it. Now, the benefits of sleep are things like maintaining a healthy weight, lowering stress levels, improved mood, increased motor coordination, as well as many benefits related to cognition and memory formation. So even though sleep feels like it's time-consuming and taking away from the world, it's really necessary in order for us to maintain a healthy lifestyle. Now, cognitive psychologists study brain waves during sleep. We can look at the brainwave activity changes and how that changes across the different stages of sleep. They do this visualization using an EEG. There are 3 types of brainwaves. The alpha brainwave, this is relatively low frequency, relatively high amplitude, and synchronized. You can see an example of alpha waves here in stage 1. Now, theta waves are low frequency and low amplitude. You can see those here in stage 2. And then delta waves are low frequency, high amplitude, and desynchronized, which you can see here in stage 3. When we are awake and alert, our brain emits beta waves, right? So you are listening to this lecture and your brain is emitting beta waves. The beta brain waves are rapid with a high number of cycles per second. So that's what you see up at the top. This indicates frequent impulses of electrical activity in the brain. When we are awake yet relaxed, our brain emits alpha waves. So alpha is awake but relaxed. As we move forward into each of the individual stages of sleep, different brain waves can be seen on an EEG. As we go through the different stages of sleep, starting with stage 1. Stage 1 is a transitional phase, occurring between wakefulness and sleep. Your respiration rate and your heartbeat begin to slow down. Your overall muscle tension and core body temperature decrease. And this is where you begin to see those alpha waves. During stage 2, the body goes into deep relaxation. Theta waves begin to show on an EEG. In this stage, you also see sleep spindles, rapid bursts of high-frequency brain waves, and K complexes, which are very high amplitude patterns of brain activity. So say you're on the couch watching television and you begin to fall asleep. This is when your body goes into deep relaxation mode. But you may still hear what's on the television, even though your eyes may be shut. So your brain is still responding to what it might be hearing. Then in stages 3 and 4, this is known as slow wave sleep. This is when you begin to see delta waves. And your respiration and heart rate slow down even further. Now in REM sleep, this is when rapid eye movement occurs. Just after stages 3 and 4, you begin to have rapid eye movements, paralysis of voluntary muscles, and dreams occur during this stage. And your brain waves are similar to those seen during wakefulness. Now, if we look at this hypnogram of sleep stages, you can see someone is awake, and then within the first hour, they dip on down into stage 3, and then they might cycle back up and back down, and then back up again. Now, I mentioned that during the REM cycle, this is when dreams occur. There are many theories on the meaning of dreams that vary across different cultures and periods of time. There is a most common dream plot. Can you guess what that is? Many times, dreams can explain a variety of different things. The first to theorize around dreams was the Freudian theory from Sigmund Freud. His is the most controversial and best known theories of dreaming. In his book titled Interpretation of Dreams, Freud called dreams the royal road to the unconscious. According to Freud, dreams allow us to express fears and sexual and aggressive desires without the censorship of our conscious thought process. Having straightforward dreams about these unacceptable desires would cause us anxiety. Instead, we dream in symbols that represent our unconscious desires. For Freud, dreams contain both manifest content, this was the actual content of the dream, which is what you recall when you awake, and the latent content of the dream, the hidden meaning of the dream. This is the hidden or symbolic meaning. These are often driven by conscious-unconscious conflicts. So the manifest content of your dream might be, I was being chased by a crab. But the latent content would show that there's a hidden meaning behind what that actually means. Now, Jung believed that dreams allowed us to tap into the collective unconscious. He believed that the collective unconscious It was a theoretical repository of information shared by all people across all cultures. He believed that certain symbols in dreams reflected universal archetypes. The research around dreams is that dreams may represent life events that are important to the dreamer. Dreaming may represent a state of proto-consciousness or a virtual reality in the mind that helps a person during consciousness. Lucid dreams are certain aspects of wakefulness that are maintained during a dreaming state. A person becomes aware that they are dreaming when they're in a lucid dream. We study insomnia during the consciousness chapter because it's defined by difficulty falling or staying asleep for at least 3 nights a week for at least 1 month's time. This is the most common sleep disorder. It may be associated with symptoms of depression. Contributing factors include age, drug use, exercise, mental status, and bedtime routines. The treatment for insomnia is stress management techniques, changes in problematic behaviors that could contribute to insomnia, and cognitive behavioral therapy, which focuses on cognitive processes and problem behaviors. Not everybody sleeps soundly. An estimated 50 to 70 million adults in the US experience a sleep disorder or disturbance in the normal pattern of sleep. Sleep disorders also affect approximately 25 to 40% of children and adolescents. Insomnia is the most common reported sleep disorder, with more than a third of American adults reporting insomnia in the last year. Insomnia can be treated medically using anti-anxiety or sedative medications, such as Xanax or Ambien, or by taking over-the-counter medications such as Somanex or Nytol that contain antihistamines and pain relievers to induce sleepiness. However, long-term use of these drugs may lead to dependence and serious side effects, including memory loss, fatigue, and increased sleepiness. Now, parasomnia involve unwanted motor behavior and experiences throughout the sleep cycle. This includes sleepwalking, REM sleep behavior disorder. So this occurs when the muscle paralysis associated with REM sleep does not occur. Restless leg syndrome involves uncomfortable sensation in the legs when they try to fall asleep that are relieved by moving the legs. And night terrors. Witnessing someone who's having a night terror is not fun. Now, sleepwalking, people with this disorder get up and walk around during deep sleep, sometimes performing actions that make them appear to be awake. They may cook, eat, open doors, or engage in minimal conversation. Because sleepwalkers are asleep, they may injure themselves or others. In restless leg syndrome, symptoms only occur primarily at night. This typically affects 5% to 25%, and the incidence is higher in women. And the cause of this restless leg syndrome is unknown. The current hypothesis focuses on genetic components, low levels of iron in the brain, and the role of neurotransmitter dopamine, as dopamine is needed to produce smooth muscle activity and movement. Now, in night terrors, during night terrors, people awaken in an apparent state of fear. Their heart rates and breathing are rapid, and they may scream loudly and sit up in bed, wide-eyed and with terror. People who have night terrors rarely recall the incident in the morning, although they can occur any time in one's life. Night terrors are more commonly reported in children between the ages of 4 and 12. Sleep apnea is becoming a sleep disorder that's more common. It's a disorder in which a person stops breathing while sleeping. It occurs when individuals, usually for 10 to 20 seconds or longer, and it's repeatedly disrupting their sleep, leading to increased levels of fatigue. Now, there are two types of sleep apnea. Obstructive, which is the airway becomes blocked and air is prevented from entering the lungs, or central. This is when the central nervous system fails to initiate breaths. Treatment can include a continuous positive airway pressure, or CPAP device, which pumps air into the person's airway. I'm skipping one of the slides. I don't love to cover this particular slide, so I'm going to skip to narcolepsy. This involves an irresistible urge to fall asleep during waking hours, often triggered by states of heightened arousal or stress. This shares many features of REM sleep, including cataplexy, so loss of muscle tone while awake, or in some cases, complete paralysis. They also experience a type of hallucinations. These are vivid, dreamlike hallucinations. Someone with narcolepsy, the usual treatment for this is psychomotor stimulant drugs. Now, when talking about consciousness, sleep is an important part of it. However, we also need to discuss substance use disorders when talking about consciousness. Substance use disorders is a compulsive pattern of drug use despite negative consequences. This is according to the Diagnostic and Statistical Manual of Mental Disorders. It involves physical and psychological dependence. Physiological dependence involves changes in normal bodily functions and withdrawal upon cessation of use. Psychological dependence is an emotional need for the drug. Question 3 is around tolerance and withdrawal, or around tolerance and addiction. It's going to ask you to define what tolerance is, which you see here, and it occurs when a person requires more and more of a drug to achieve the effects previously experienced at lower doses. And it does have a linkage to physiological dependence. Now, addiction is a need for the drug in order to function in daily life. That's how those two things differ. You're going to be asked to figure out a way to test for those two things. How can you test if someone has a tolerance or if someone has an addiction? I recognize that that could be a difficult question, so I would really recommend using outside sources, but just make sure you cite those sources appropriately. Now, withdrawal is negative symptoms experienced when drug use is discontinued, which oftentimes, if an addiction exists, you'll likely see withdrawal when the drug is discontinued. Now, different drug categories. Many of times, these categories overlap. For example, you have depressants, things like alcohol, barbiturates, opium, and things like marijuana. But those also tend to be hallucinogenics. Nicotine is both a stimulant and a depressant. You have insane amounts of drugs that could potentially affect many things. So you have stimulants. These are things like cocaine and amphetamines. These will increase your heart rate and blood pressure and body temperature. Okay, so stimulants increase things. When used, it increases your alertness. You experience mild euphoria. You have a decreased appetite in low doses. In high doses, you have increased agitation and paranoia. It can cause hallucinations. Some can cause heightened sensitivity to physical stimuli. Some doses of MDMA can cause brain toxicity and even death. It is psychologically addicting. All of these are. Depressants or sedative hypnotics are things like alcohol and barbiturates, benzodiazepines. The reason they're called depressants is because they decrease your heart rate and blood pressure. So they create relaxation, decreased inhibitions. High doses can induce sleep, passing out, cause motor disturbance, memory loss, and decreased respiratory function. Now, opiates like heroin and fentanyl and morphine and oxycodone, they also decrease pain, pupil dilation, decreased gut mobility, and decreased respiratory function. But they're involved in pain relief, euphoria, sleepiness. High doses can cause death. And hallucinogens, things like marijuana and LSD, increase our heart rate and blood pressure, and they may dissipate over time. If we look at depressants more clearly, these are drugs that suppress our central nervous system activity. Usually GABA agonists, which have a quieting effect on the brain. They work by binding to GABA receptors, which make the neuron less likely to fire. Alcohol, specifically as a depressant, decreases reaction time and visual acuity. Think about watching a show, seeing someone who might be drunk, and their inability to catch something when someone tosses something their way. They have lower levels of alertness. Takes you a while to catch their attention. Reduces behavioral control and can result in complete loss of consciousness. Now, stimulants, however, increase our overall lack of neural activity. Usually dopamine agonists, which work by preventing the reuptake of dopamine. Dopamine activity is associated with reward and craving. Therefore, these drugs can be highly addictive. Side effects can include nausea, elevated blood pressure, increased heart rate, feelings of anxiety, hallucinations, and paranoia. This is a visual of your dopamine agonists and the dopamine reuptake when taking a stimulus. Now you have nicotine and caffeine. These are also stimulants. However, they're a little bit more socially acceptable. Caffeine involves antagonizing endozyme activity. So it allows for increased levels of alertness and arousal. Now, nicotine interacts with our acetylcholine receptors. It's highly addictive and plays a major role in arousal and reward mechanisms. Opioids serve as analgesics, a decrease in pain, through their effects on the endogenous opioid neurotransmitter activity. These also are highly addictive. Hallucinogens cause changes in sensory and perceptual experiences, can involve vivid hallucinations, variable with regards to the specific neurotransmitter systems they affect. Now, other states of consciousness that we want to discuss are things like hypnosis. Hypnosis is an extreme focus on the self that involves suggested changes of behavior and experience. If any of you have ever participated in a hypnotist event, it can be wild. Clinicians might actually use hypnotism in an attempt to alter the thoughts and perceptions of a patient. It has been used to draw out information believed to be buried in someone's memory. So you might use a hypnotist technique in order to retrieve a memory that someone may have kept deep and dark and hidden. Some clinicians might use hypnotism to help people stop smoking or overeating. Unlike portrayals in the media, individuals undergoing hypnosis are in control of their own behaviors. And people vary in their ability to be hypnotized. Uses include pain management, treatment of depression and anxiety, quitting smoking, and weight loss. That's why when you're at an event where there's a hypnotist, they take them up and they do an exercise first to see how well they can be hypnotized. And if they're not going to be able to be hypnotized, they can tell and they can release them back to the audience. Another state of consciousness is meditation. This is the act of focusing on a single target, such as breath or a repeated sound, to increase awareness of a moment. Meditation involves relaxed yet focused awareness. It shows promise in stress management, sleep quality, pain management, and treatment of mood and anxiety disorders. 2 other questions for chapter 4 that I didn't cover during the lecture is a question on introspection. Introspection was used by William James when first beginning to study consciousness. Those questions I asked you at the beginning of the lecture, that was introspection. That was showing you introspection. The question is going to ask you to define what introspection is, but then also to talk about how it fell from favor as a means to studying consciousness. Why didn't it work? And I want you to talk about the fact that there's no scientific basis. There's no way to test introspection as a valid way to study consciousness. Then your 4th question is about relapse. You may need to use an outside source. Your book also talks about relapse, but you want to talk about relapse and other ways that you can prevent relapse and the probability of relapse in drug abusers. Next is chapter 5, so sensation and perception. In order to study sensation and perception, I want you to think about a time that you've had the experience of watching an event with others. This is the sensation. And then being surprised by the different interpretations of what we hear, of what just happened. This is perception. You may all be experiencing the same sensation. You're all at the same event, but how you perceive those things all differs based upon your perception. Our sensory systems are responsible for providing information about our surroundings, which allow us to successfully navigate and interact with our environments. If you were standing in the midst of the street scene above, you would be absorbing and processing numerous pieces of input. Sounds, sights, and auditory senses. In order to understand sensation, we need to understand our sensory receptors. These are specialized neurons that respond to specific types of stimuli. And to define what sensation is. Sensation occurs when our sensory receptors detect a stimuli. So as soon as our eyes detect a visual stimuli, our ears detect a sound, our mouth detects a taste, our hands, our body, our skin detects touch, and our nose detects a smell. When sensory receptors detect a specific stimuli, they convert that energy into an action potential, which is sent to our central nervous system. This is called transduction. Now, each one of our 5 senses is technically a sensory system. Our visual sensory system helps with balance and vestibular sense. Our hearing or auditory system is body position. You may turn towards a noise. Your smell or olfactory system helps in movement. Your taste and gustation helps with pain. And touch or somatosensation system helps with temperature. Now, if we break sensation down even further, for a stimulus to cause an action potential, meaning for one of those sensory receptors to actually create an action in us, a response, it first has to be strong enough to be detected. This is known as absolute threshold. This is the minimum amount of stimulus energy that must be present for the stimulus to be detected half of the time. Some typical thresholds. So for vision, seeing a candle flame 30 miles away on a dark, clear night. For hearing, the equivalent of the ticking of an expensive watch 20 feet away. For olfaction, or smell, a drop of perfume in the air filling a 6-room apartment. For taste, the equivalent of 1 teaspoon of sugar in 2 gallons of water. Touch, the equivalent of feeling the wing of a fly fall on your cheek from a distance of 1 centimeter. Now, we are also able to receive messages presented below the threshold of conscious awareness. These are known as subliminal messages. The stimulus causes an action potential, but we're not consciously aware of it. Now, a just noticeable difference, or JND, is the minimum difference in stimuli required to detect a change or a difference between stimuli. This can change depending on the stimulus intensity. A cell phone lighting up in a dark movie theater is more likely to be noticed than if it lights up in a brightly lit shopping mall. The brightness of the cell phone does not change, but the ability to detect the change in illumination does. Now, if we switch to perception, this is our sensory receptors collect all the information about our environment. So, those 5 sensory systems: vision, taste, touch, smell, vision, taste, and hearing. But it is then how we interpret that information that ultimately affects how we interact with the world. Perception is defined by a way that sensory information is interpreted, organized, and consciously experienced. There are 2 types of perception. You have bottom-up processing. This is a system in which perceptions are built from sensory input. So you're taking that sensory input and you're building perceptions upon that input. Top-down processing is interpretations of sensations, is influenced by available knowledge, experience, and thoughts. So you're perceiving something based upon already preconceived notions without the senses. You can look at it from this example. So top-down occurs when previous experience and expectations are first used to recognize the stimuli. It helps you to rapidly organize features into a meaningful whole. In bottom-up processing, we are sensing basic features of stimuli and then integrating them. So we're starting with the raw materials. That is, we begin with the small sensory units and build upward to a complete perception, regardless of what was previously interpreted. Now, there are some factors that influence our perception. You have sensory adaptation, not perceiving stimuli that remain relatively constant over prolonged periods of time. For example, when you first enter a quiet room, you might hear the clock ticking, but over time you become unaware of it. The ticking is still affecting sensory receptors, but you're no longer perceiving the sound. It's no longer a part of your world. And the second thing affecting is attention. Our ability to give attention to is important. An energy activates our sensory receptors. We use attention to determine what to focus on and what to disregard. There are 3 important characteristics of attention. Attention improves our mental processing. It takes effort, but it's also limited. We rarely have the luxury of paying attention to any single stimulus, and we simply cannot process all the information converging simultaneously into our sensory systems. In order to prioritize so that we don't become overloaded, we have to use selective attention, or the ability to focus on a subset of available information and exclude the rest. Selective attention can be described as a filter that blocks certain information from reaching your awareness. At times, we can even suffer from inattentional blindness. This is a failure to notice something that is completely visible because of a lack of attention. Nearly 1/3 of participants in a study did not notice that a red cross passed on the screen because their attention was focused on the black or white figures. The third thing that affects perception is motivation. Sometimes we think we hear something, such as a phone ringing, when it is not, because we are motivated to perceive it. We are waiting for an important phone call, so anything we hear ringing, we're going to perceive as a phone call because we're motivated to hear that. This is also known as signal detection theory. This is a change in stimulus detection as a function of current mental state. Where you're at mentally can affect how easily you detect a stimulus. Now, the fourth thing affecting our perception is our beliefs, values, prejudices, and expectations. People who hold positive attitude towards low-fat foods, for example, are more likely to rate foods with low-fat labels as tasting better than people with less positive attitudes about low-fat foods. How you perceive things is also affected by your beliefs, values, prejudices, and experiences or expectations. You then also have to think about life and cultural experiences when you're perceiving something. One study found that people from Western cultures, where there is a perceptual context of buildings with straight lines, think of all the cities with all of our skyscrapers, we're more likely to experience certain types of visual illusions, like the Muller-Lyer illusion, than individuals from non-Western cultures, where they may live in round huts, for example. Here is the Muller-Lyer illusion. Lines appear to be different lengths, although they are identical. Arrows at the ends of lines may make the line on the right appear longer, although the lines are the same length. And when you apply these lines to a three-dimensional image, the line on the right again may appear longer, although both black lines are the same length because of how far away they are on that 3D image. Now, perception, when it comes to visual and auditory stimuli, both occur in forms of wavelength. Two physical properties of waves are amplitude and wavelength. The amplitude or height of a wave is measured from the peak to the trough. The wavelength is measured from peak to peak. Now, the frequency of both visual and auditory waves is the number of waves that pass a given point in a given time period. Longer wavelengths have lower frequencies, and shorter wavelengths have higher frequency. Now, if we look at this directly when concerning light waves, the visual spectrum is the portion of electromagnetic spectrum that we can see. Okay? Very small amount. Different species can see different portions of the spectrum. Humans can see wavelengths ranging from about 380 to 740 nanometers. And how we perceive color depends on both wavelength and amplitude. Different wavelengths of light are associated with our perception of different colors. Longer wavelengths are equal to reds, intermediate wavelengths is greens, and shorter wavelengths is blue. And the amplitude of light waves is associated with their brightness and intensity, so a larger amplitude appears brighter than a lower amplitude. Now, if we take frequency and amplitude and look at it from a sound wave, the frequency of sound waves equals the pitch. So if it's a high frequency, it's a high pitch sound. Low frequency wavelengths, it's a low pitch sound. The audible range of sound frequencies in humans is between 20 and 2000 hertz. And the amplitude of a sound wave equals the loudness. The higher the amplitude, the louder the sounds. The lower the amplitude, the quieter the sound. We measure loudness in decibels, and a typical conversation occurs at about 60 decibels. A rock concert occurs around 120 decibels, but the potential for hearing damage is between 80 and 130 decibels, and the threshold for pain is 130 decibels. And if we think about sound waves on a spectrum and their sound intensities, you can get the loudness of common sounds from some of these things. So you've got a whisper just above 20 decibels. Soft music is about 40 decibels. A conversation is around 60 decibels. A loud car is around 80 decibels. A blender is just above 80 decibels, probably somewhere around 90. And a loud rock concert is around 120 decibels. Now, what we're going to do is go through each of the visual systems so that you can understand the anatomy behind the visual systems. Not just the visual system, but the auditory system, taste, touch, all 5 of your senses. We're going to go through the anatomy of each of these systems. So first up is the anatomy of the visual system. Light waves are transmitted across the cornea and enter through the pupil. The pupil size is controlled by muscles that are connected to the iris. This is your colored part of the eye. The light crosses the lens, is focused on the fovea, which is part of the retina. The fovea contains your photoreceptors. The photoreceptors are connected to retinal ganglion cells. These are axons from these cells. Exit through the back of the eye, where they form the optic nerve. And the optic nerve then carries the visual information to the brain. Now, everyone has a blind spot. This is a point where there are no receptors, where information exits the eye. So, where the optic nerve is in your eyeball is actually where your blind spot is, and where we cannot respond to visual information. If you look at it a little bit deeper, you can see it here. Now the photoreceptors, inside of the photoreceptors are cones and rods. Cones help with daytime vision. They work best in bright light conditions. They have high acuity to color information and are located in the fovea. Now rods are for your nighttime vision. They work best in low light conditions. They have a very high sensitivity. Think about when you're driving at night and headlights are coming towards you and you instinctively want to cover your eyes because it's so bright. That's because your rods are at work here. They allow for low acuity vision in dim light. They're involved in the perception of movement in our peripheral vision, and they're located in the periphery of the retina. Now, given what you know about rods and cones, which would you prefer to live without? Cones help you to see color and differentiate between color information, whereas rods allow you to see at night. Could be interesting to think about which one you prefer. Now in the optic chiasm, the optic nerve of each eye merges at the optic chiasm. This is an X-shaped structure just below the cerebral cortex. Information from the right visual field is sent to the left hemisphere, and information from the left visual field is sent to the right hemisphere. So what your right eyeball is seeing is actually broken down by and processed by the left side of your brain. And what's taken in by the left eyeball is done by the right. Both of which then converge in the occipital lobe for processing. Your occipital lobe is what's responsible for processing the information and understanding what it's seeing. Now the visual pathways. After being processed in the occipital lobe, visual information exits and goes through 2 different pathways. The what pathway, so object recognition and object identification. Do I recognize what I'm seeing and can I identify it? It also then goes through the where-how pathway. Where is it located in space? And how one might interact with a particular visual stimulus. How do I interact with what I'm seeing? Now, color vision happens through a few different theories. You have the trichromatic theory of color vision, meaning that all colors can be produced by combining red, green, and blue. This applies to the retina, where color vision is controlled by 3 types of cones. Or it's thought to be looked at from the opponent process theory. Color is coded in opponent pairs: black and white, yellow and blue, and green and red. Some cells are excited by one of the opponent colors and are inhibited by the other. And it applies to cells after the retina. Now, research has found that both theories are true, but for different parts of the visual system. Now, if we want to look at support for opponent process theory, if you stare at a colored stimulus, the colored pairings of the opponent process theory lead to a negative afterimage. So if you stare at the white dot for 30 to 60 seconds, now move your eyes to a blank piece of white paper or a blank wall. What you should see is an afterimage. The exact replica of the image you see there should display on that white piece of paper. So this exercise supports the opponent process theory. Now, depth perception is our ability to perceive spatial relationships in 3D. Depth cues of a visual scene are used to establish our sense of depth. Depth perception is incredibly important to everyday activities. It's what allows you to kick a soccer ball appropriately at the appropriate time. Do a cartwheel and make sure to put your hands down at the right time. Now, there are depth cues. You have binocular cues. This is a cue that relies on the use of both your eyes. You have a binocular disparity, a slightly different view of the world that each eye receives. And you can see this by covering one eye and then covering the other eye. What you see ends up being different. You also have monocular cues. These are cues that rely on only one eye. So you have a linear perspective when 2 parallel lines seem to converge, like you see in this picture to the right. And interposition, the partial overlap of objects. You can see this convergence of these 2 horizontal parallel lines with just one eye. You don't need both eyes in order to see that. Color blindness is another visual system that affects a lot of people. Color blindness is the inability to distinguish 2 or more shades in the color spectrum. And there are different kinds of color blindness. You have monochromats who have total color blindness. Their worlds look black and white. This kind of color blindness is rare and results from individuals having only rods or only one kind of functioning cone instead of 3. Dichromats usually have trouble distinguishing red from green because they have just 2 kinds of functioning cones. This is an inherited genetic defect that results in seeing mostly shades of blue and yellow, which differ in severity. Between 7 and 10% of males and about 0.4% of females have a form of red-green color blindness. The reason that males are more affected than females is that the genes for the pigments used by red and green cones are located on the X chromosome, making the red-green color blindness a sex-linked condition. Now, the next system is the auditory system. So think about your ears. If we look at the ear, it's divided into 3 divisions. The outer ear, which is the pinna and the tympanic membrane. The pinna is earlobe, essentially. The middle ear is the three ossicles, the malleus, the incus, and the stapes. And the inner ear is the cochlea and basilar membrane. You can see all of those systems here in this visual. The external ear, the middle ear, and the inner ear. Now, the steps of auditory transduction. Sound waves travel along the auditory canal and strike the tympanic membrane, which causes your ear to vibrate. The vibration causes the three ossicles to move. This presses the stapes into the oval window of the cochlea. Fluid inside the cochlea begins to move, stimulating the hair cells, which are your sensory receptors for sound. The hair cells generate neural impulses that travel along the auditory nerve to the brain. The auditory information is sent to the inferior collicus, then the medial geniculate nucleus of the thalamus, and finally to the auditory cortex in the temporal lobe for processing. Now, how does the auditory system differentiate among various pitches? Two theories. First is the temporal theory. Frequency is coded by the activity level of a sensory neuron. The problem with this is the frequency of action potentials cannot account for the entire range that we're able to hear. There is a point at which a cell cannot fire any faster. However, place theory is where different portions of the basilar membrane are sensitive to sounds of different frequency. The base responds to high frequencies and the tip responds to low frequencies. Both theories explain different aspects of pitch perception. Then localizing sound involves the use of two cues. Monaural cues, which is one ear. Each ear interacts with incoming sound waves differently. Or binaural cues, or two ears. Provide information on the location of sound along a horizontal axis. Relies on difference in patterns of vibration. Now, interaural level difference is sound coming from one side of the body is more intense at the closest ear because of the attenuation of the sound waves as it passes through the head. And the interaural timing difference is small difference in the time at which a given sound wave arrives at each ear. Now, hearing loss can affect the auditory system. Things such as deafness. This is the partial or complete inability to hear. You then also have congenital deafness. This is deafness from birth. Conductive hearing loss is associated with a failure in the vibration of the eardrum and or movement of the ossicles. This can often be dealt with by using hearing aids which amplify incoming sound waves to make vibrations of the eardrum and movement of the ossicles more likely to occur. Conductive hearing loss can be caused by age, genetic predisposition, environmental effects. So exposure to extreme noise, certain illnesses or damages due to toxins. So think about if you live and grew up in a home that was right near a set of train tracks where trains were constantly driving by and creating a loud environment and extreme noise. This could potentially cause conductive hearing loss. Sensory neural hearing loss is a failure to transmit neural signals from the cochlea to the brain. Can be caused by Meniere's disease, which results in degeneration of inner ear structures. Here are some examples of environmental factors that could influence conductive hearing loss. Now, taste. Research has demonstrated that we have about 6 groupings of taste or gustation. Sweet, salty, sour, bitter, umami. And some research suggests that we process a taste for the fatty content of food, which means that the sixth one is probably some likely fat content. The taste buds on your tongue are groupings of taste receptor cells with hair-like extensions that protrude into the central pore of the taste bud. Could you imagine that you actually have teeny tiny little hairs on your tongue? Each taste bud has a life cycle of 10 days to 2 weeks, which means that your tongue is constantly rejuvenating your taste buds. Now, transduction of taste has only a 2-step process. Taste molecules bind to receptors and cause chemical changes within the sensory cell. These changes result in neural impulses being sent to the brain. You can look at it this way. Taste buds are composed of a number of individual taste receptor cells that transmit information to nerves. This micrograph shows a close-up view of the tongue surface. See how there's tiny little holes on the tongue surface? This is a taste pore. And then below that is the taste receptor cell. Wild. Now, smell and olfactory receptor cells contain small hair-like extensions as well, which serve as the site for odor molecules to interact with chemical receptors located on these extensions. And transduction of odor molecules happens when odor molecules bind to these receptors. Chemical changes cause signals to be sent to the olfactory bulb, where the olfactory nerves begin. And information is sent to the limbic system and primary olfactory cortex. When talking about smell, we need to discuss pheromones. These are chemical messages sent by another individual. Many species respond to pheromones sent by another individual, usually communicate information about the reproductive status of a potential male. And you see those more being used with animals. Now, touch. There are many types of sensory receptors located in the skin, each attuned to specific touch-related stimuli. So you have Meissner's corpuscles. These respond to pressure and low-frequency vibration. Pacinian corpuscles detect transient pressure and high-frequency vibrations. Merkel's discs respond to light and pressure. And Ruffini's corpuscles detect stretch. Thermoception and nociception. As well as receptors located in the skin, there are a number of free nerve endings that have sensory functions. Those functions are temperature perception, or thermoception, and sensory signal indicating potential harm and maybe pain. This is nociception. This type of sensory information travels up the spinal cord directly to the brain, specifically the medulla, thalamus, and the somatosensory cortex. Pain perception is important because it motivates us to remove ourselves from the cause of injury. You have a question called no pain, no gain, where you may hear someone say that they would rather never experience the pain associated with a significant injury ever again, and they would do anything. But how would you respond to them given pain perception and its importance to help motivate us to remove us from things that could cause serious injury? Inflammatory pain signals some type of tissue damage. Neuropathic pain caused by damage to neurons of either the neuroperipheral or central nervous system. And congenital insensitivity to pain is a rare genetic disorder in which the individual is born without the ability to feel pain, can detect differences in temperature and pressure, but cannot experience pain. These individuals who cannot ever experience pain are at a greater risk of injury and have shorter life expectancies because of it. Now, the vestibular sense within the ear is our ability to maintain body posture. The major sensory system, vestibular system, and the cochlea in the inner ear. These organs allow for us to be able to stand at attention and maybe even pick up one foot and stand on one leg and maintain our bodily posture. The maculae are specialized for sensing linear acceleration, such as when gravity acts on the tilting head, or if the head starts moving in a straight line. The difference in inertia between the hair cells and the membrane in which they are embedded leads to a shearing force that causes the bend in the detection of linear acceleration. The vestibular system also collects information important for controlling movement and reflexes that move parts of our bodies to compensate for changes in body position. Both the proprioception and the kinesthesia interact with information provided by the vestibular system. Proprioception is the perception of body position, and kinesthesia is the perception of the body's movement through space. Information from the brain is sent to the sensory organs in a proprioceptive and kinetic systems. Now, one of your questions this week is around in the moment. I want you to describe all of the sensations that you're aware of at this moment. What happens to your description as your attention shifts from sense to sense? So I want you to go through each of your sensations, each of the 5 sensations, your taste, your smell, your hearing, your vision, and I'm forgetting the last one. Taste, sound, vision, hearing. No, taste, smell, touch. So the 5 systems: touch, taste, smell, hearing, vision. Then I want you to rank and tell me which one is the most important sense to you and why. Okay? So that question, you need to go through each of your 5 senses and say what they're sensing in that moment as you're answering this question. And then you need to rank the most important one. Now, all of sensation and perception is around the Gestalt principles of perception. Gestalt psychology is a field of psychology based on the idea that the whole is different from the sum of its parts. Our brain creates a perception that is more than simply the sum of available sensory inputs. The brain does this in predictable ways, which Gestalt psychologists translated into principles by which we organize our sensory information. These principles include figure-ground relationship, proximity, similarity, continuity, and closure. So if we look at the figure-ground relationship, the idea is that we tend to segment our visual world into figure and ground. Figure is the focus of the visual field. Ground is the background. Our perception can vary depending on what we view as ground and what we view as figure and what we view as ground. If you look at the image here on the slide, the figure-ground relationship is explained. You can either view it as a vase or a pair of faces, depending upon which thing you view as the figure and which thing you view as the ground. If the black area is the background or the ground, and the white area is the figure, then you're focusing on the white area and you think, oh, a vase or a vase. But if you switch that and the white area becomes the background or the ground, and the black area becomes the figure, you see the outline of 2 faces in profile. Weird faces, but faces nonetheless. Now, the principle of proximity. The idea that things that are close to one another tend to be grouped together. In proximity, This would suggest that you see one block of dots on the left side, so figure A is one series of dots, and figure B is 3 columns, 3 different groups of dots, merely because of the proximity that they are to one another. You then have similarity. The idea that things are alike tend to be grouped together. When looking at the array of dots, we likely perceive alternating rows of colors. You can see 3 rows of blue and 3 rows of yellow, not just 6 rows of dots. You then have continuity. This is the idea that we are more likely to perceive continuous, smooth flowing lines rather than jagged, broken lines. Good continuation would suggest that we are more likely to perceive this as 2 overlapping lines, rather than 4 lines meeting at the center, because of their continuity. You then have the principle of closure. This suggests that we perceive a complete circle and rectangle rather than a series of segments. If you take a look at the following figure, how might you influence whether people see a duck or a rabbit? If you keep it in this form or shape, it looks very much like a duck. But if you rotate the photo to the right, you might then begin to see a rabbit. Now, your perception is built from senses and influenced by experiences, biases, and culture, as we discussed earlier. Dr. Atiba Goff's research showed that Black boys were considered more responsible for their actions than white boys of the same age. 264 university students, mostly white females, involved in the study. As a group, they overestimated the age of Black children by 4.5 years, so a Black 12-year-old male may seem 15 or even 16 years old. Also judged Black children over the age of 10 as significantly less innocent than white children of the same age. What might this say about law enforcement, educational, and jury perceptions of defendants of different races? The link there that's included is a link to the study at the APA, if you wanted to take a look at it. It shows very well how our implicit bias can affect our perception. The last part of this lecture will help you with question 4 for chapter 5. The central tenet of Gestalt psychology is that the whole is different from the sum of its parts. What does this mean in the context of perception? So talk about that particular section. What does it mean in the context of perception when we think about the phrase, the whole is different than the sum of its parts? And then your first question is kind of a fun one. It's about music and lyrics. Have you ever listened to a song on the radio and sung along only to find out later that you have been singing the wrong lyrics? Once you found the correct lyrics, did your perception of the song change? Oftentimes, we sing whatever it is that we hear at first rate, and we may even continue to sing it that way, even though we know it's incorrect. But how does your perception of a song change once you know the correct lyrics? Please give me an example of a specific song. All right. Additionally, this week, you also have your first exam. So your questions can be found here. Click the blue link to open up the Word document. Like I said last time, type into that Word document, save it on your computer so that you have it, and then click into the red link in order to submit the Word document with your submission. Now your exam. It has a time limit of 8 hours and 20 minutes. I recommend doing it in one sitting. It's open note and open book, which means that you can have the PowerPoints open for them. You can have any notes that you might take during my audio lectures. You can even have your textbook open when you're going to take this. And it allows for 2 attempts for the highest grade possible. Each question shows once. It allows you to move on to another question and backtrack. So you will be able to move back and forth, okay? But you can check each and then hit save answer. And it's saved, and then you can move on to the next one. Okay? Then at the very end, it tells you what your score is. And then you can even check question completion status to see which ones you've completed and which ones you need to go back and redo if you didn't complete it. Because say you're not sure about this one and you don't want to move on to the next one, so you move on to the next without it being done, and it takes you to question 3. Okay? If you have any questions at any point, please don't hesitate to reach out to me by email nwest@westfield.ma.edu. Have a great week.